I am Patrick Serruys. We are here in Seoul at TCT Asia, and I have in the panel from left to right Chuck Simonton, Tullio Palmerini, and Takeshi Kimura. And this panel of interview is made of two parts. One is about the deaths, and the second part is about the new BRS. And the rule of engagement is that we have received a few slides. We will look at this slide and then make some comment. The slides are just to trigger the reaction of the panel on the different questions. So if I may have the first slide. So as I said, there is two parts. One which is the contemporary deaths, evolution of the technique, efficacy and safety of contemporary deaths, clinical of lesion-specific death preference, and then the next generation. And I think this first slide came from the work of Robert Byrne in the Lancet, illustrate a few things, illustrate that the diameter, the thickness of the struts has decreased with time. That's certainly one point. Some of them have changed their morphology. Instead of being quadratic, they are circular. And the key point is the change in the alloy. And the change in the alloy is basically to increase the tensile strength, the radial force, and to allow to reduce the diameter. Chuck, do you believe that this is a key point in the progress and advancement of the drug-eluting stent?
Yes, and I think we've seen that from the preclinical studies that show the speed of endothelialization and healing is basically determined by the obstruction to endothelial coverage, and that's directly related to strut thickness. And actually, Julio Palma showed us that many years ago with the different heights of barriers to endothelial growth, and I think 100 microns was the threshold where you started to see a lot faster endothelial growth. I think the thinning of the struts has been an important aspect of DES and healing.
Tullio, is there a minimum? Are we going to get a plateau beyond which the radial force will not be sustainable to counteract the recoil of anything else?
Well, actually, I think that 70 micron is now the strut thickness of the new generation drug-eluting stents, which have proved safety and efficacy in the randomized trial that have been performed so far. So we really don't know if we can go further down, but I doubt it.
You have an Orsiro with 60 micron now, which is certainly quite successful and safe. I don't see somebody is going to go below the 60. Takeshi, do you believe that somebody is going to be below the 60?
I'm not so sure, but Orsiro seems to have relatively better result as compared with Zions, but the difference is mainly limited to the peri procedure myocardial infarction. So I think the further effort to further minimize the strut size would give us a much opportunity to improve clinical outcomes.
Chuck, if you had to stratify of given hierarchy, is that the thickness of the struts, the release of the drug, the shape and the design of the struts, the form of the struts? What is for you the most important thing? I mean, basically these people has reduced the diameter of the struts, and we know why, but you believe it's the key factor?
I think the thickness, not so much the width, but the thickness of the struts, which is in the flow column, plays a difference. And as I mentioned, on the speed of endothelial coverage. But the two other major components of DES, which have evolved over time, are the polymer coating and, of course, the drug. And I think most companies more or less have converged on the pharmacokinetics of drug elution being in this 90 to 120 day period of full drug elution, and everyone's kind of settled on that. You don't see a lot of change in that. There's one particular company that's working on a little longer elution rate, but other than that, everybody's converged on that. The polymer is very important because, and maybe we're going to get into that in more of your discussion, but the early polymers on the first generation drug-eluting stents had some issues. And the Taxus polymer had kind of stuck to itself when it was in contact with itself, and that created some webbing, and that was some issues that we had to deal with. The polymer on the Cypher drug-eluting stent seemed to be the one that had more eosinophilic inflammatory types of responses. And so that kind of led the field to think, well, maybe we should go away from polymer completely and go to polymer-free drug-eluting stent or a resorbable polymer. But as you know, from on our development side, it turned out we were at Abbott Vascular about to do that until we saw the thrombosis data from a very large trial in the US called Spirit 4, where we knew that Xience would have a lower restenosis rate than Taxus, but we had no idea that thrombosis was going to be lower. And when that was statistically superior, we realized that we went back and looked at the surface. The fluorocopolymer on the Xience platform is highly thromboresistant, platelet resistant, and also inflammation.
So today, to my knowledge, you have a Chinese company called Sinomet, which released the entire drug in 28 days. Before that, we had the Endeavor with 6 days. Now they are at the minimum of 28 days. And then on the other hand, you have my stent, which goes for still 2 nanogram of this microcrystal of sirolimus embedded in the wall, 2 nanogram at 9 months. Do you see a winner there, or do you see the different strategy?
Well, I'll tell you, just from our own data, the reason we came up with the 90 to 110 day, 20 days, which Boston Scientific also converged on in Cordis in the day with the Cypher stent, was that the period of reaction to the balloon and stent injury, the acute mechanical injury of the vessel, seems to go on, the neointimal proliferation, for at least 2 to 3 months in animal models. So we felt like we needed to cover that time period. So 28 days, in my our own opinion, would be too short. But good luck, maybe they'll hit on it. And then the longer persistence of drug in the vessel wall, which is being done by the MyStent by having a crystalline structure to the sirolimus, still needs to be looked at. The clinical data so far from Dissolve 1 and 2, the trials that they've done so far looks promising. Yeah. But the event rates now clinically for patients are so low that most of these new stent platforms are showing equivalency but not superiority to where we are.
That will be the question on the next slide. Takeshi has written an editorial for the Lancet on one of our papers, the MyStent, and the title of your editorial was Non-Inferiority, Me Too. Yeah. And indeed, we can barely read the number there, but all these classic trials have a margin of non-inferiority of 6.4, 6.8, and it doesn't change too much. So, Tullio, is that the way to go? How long are we going to do non-inferiority trial?
Yes.
Is there something we should change there?
Well, actually, yes, I think this is a problem. We have too many non-inferiority trials, and non-inferiority trials have many limitations because they often mix heterogeneous endpoint, and they often are not powered to determine significant differences in important endpoints such as death, myocardial infarction, or stent thrombosis. But worse than this, many of these trials turn out to be underpowered because expected event rates were higher than observed, and this trial ended up to have a very high disproportionally high non-inferiority margin. So I think that many non-inferiority trials have commercial issues and not scientific issues. So I think we should do less trial,
So
but
the
more powered.
The Me Too
Yeah.
is justified. Is justified. You know, in the next slide, there is the question: which stent should we select for the left main? And you see a forest plot there for some of them, the cobalt chrome everolimus, and then I guess a biometrics, and then a platinum chrome synergy. At first sight, it doesn't seem that there is a real winner. What do you think, Takeshi? What is your favorite stent for the main stem?
Personally, I do not have so much preference for some specific stent for left main intervention, but the difficult point of left main intervention is not only the device itself, but also the technique is very variable and may be very important to for the outcome. So I think the very large study, and we also control some technique as well as the stent type for this type of comparison.
When you are stenting a left main, are you considering the area of the cell?
Yeah.
Is that a major criteria for you? And which one, in your experience, has a larger cell which allow an easy access either to the LED or the circumflex?
Some of the Japanese physicians are doing many left main procedures prefer a Nobori stent, which is 3.5 millimeter Nobori stent, which has a very large expansion limit and also the large cell for the side branch.
I see. Any comment? Yeah.
Yes, we have to acknowledge that these are observational data. So observational data are at best hypothesis generating. So if we want to follow evidence-based medicine, actually there are only two trials with the newer generation drug-eluting stent. One is the Excel trial, which use the Xia stent, and the other is the Nobel trial, which use the biometric stent. So I think these are the only two stents that right now have been studied in a randomized trial.
Yeah, and if you look the Biolimus A9, the biometrics is quite nicely to the left in the first column. What do you think about that question?
Yeah, I think I want to get back to what Tullio said, which is the data that was on that slide comes from Korea, and it's a terrific data set that has been created here out of Asan Medical Center by S.J. Park. But it's observational data, and none of that is head-to-head randomized trial data. It's very, very important. The only stents, as Tullio mentioned, that have really been in big left main trials versus coronary bypass surgery are the two that he mentioned, and and those are the ones that really have the evidence that you can rely on what the event rates really are, because these were very highly monitored trials and very well run studies in head-to-head versus coronary bypass surgery in complex patients. The XL trial was a left main trial, as you know, so that all the patients had to have significant left main disease, and many had complex, of course, 3-vessel disease. So these patients are complex.
So the panel has the impression that we have written a plateau for statistical reason. I mean, with the non-inferiority, you cannot go forever. In terms of dimension, except if there is another alloy even stronger than cobalt chrome or platinum chrome. So that editorial seems to say that's it, we are mature, we will not go further. I would like to show you two slides that I have had One is very recent, that's Kanzari, Laura Mori. This is the Science or Zero, initially submit to the New England and then end up in the Lancet. And as you could see, something happens early in the game, I would say on day one, at the time of the peri-procedural. And the marketing is insisting about the fact that the target lesion failure is there after 1 year. But of course, these two curves run into parallel. What is your reaction on this kind of slide? Because it seems to indicate that something is superior to the science. And as a review, I can tell you what I know about this trial, but first I'll let you say what you think about it.
Yeah. I think Tullio is probably the best qualified to talk about the design of this particular study, because it's not simply a head-to-head randomized trial. It included some Bayesian analysis, which incorporated some preceding data. So you have to be a little bit careful about how the data was put together to come up with this data set for both the Science and the Orsaro arms. And then there are some questions also, and as a reviewer, you can probably comment more specifically about the definition of peri-procedural myocardial infarction.
Right. So they use the whole definition of ARC, where when the paper come back, we were only talking about CKMB in the initial ARC, and circulation said if you take 3 CKMB, you should also take 3 troponin, and of course that was a total imbalance, and that imbalance exists from that time.
Right.
So I think there are a couple of considerations that we could make regarding this trial. So first of all, as you have said, all the difference is in the peri-procedural period. So this has nothing to do with the polymer, but probably with the platform. But there are also other consideration that I would like to do. I mean, if you see the baseline characteristic of patients treated with Xience and those treated with O0, so for some reason, patients treated with Xience had more complex coronary artery disease. They had longer stent, they had more overlapping stent, and they had also more stent per lesion. So if you correct, and they did it for all these confounding factors, actually the difference in the primary endpoint was not significant anymore. So there was a trend, but p-value was 0.06. So the other consideration I would like to do is that you don't see the same finding, for example, in the bioscience trial, which is a larger trial in which there was no difference in target vessel myocardial infarction.
Which was run already in 2014 and published in the Lancet by Thomas
Pilgrim.
Pilgrim,
And if any, I mean, rates of stent thrombosis were 66% lower in the Xience stent than in the O0. So I think, I mean, these data are very interesting, but they must be put in the
marketing department has done a very good job, let's say that. This is a little bit more, has been published by the Lancet. I mean, I was mentioning the editorial of Takeshi, and the people get somewhat excited because you see around 180 days, it seems that the science diverged somewhat from the my stent. Honestly, I can tell you, and it's a scoop, the 2 years follow-up doesn't show that anymore. But at that time, at least on target lesion revascularization, there was potentially a sign. I think we have to move to the next in the last 10 minutes, and we can go quite quickly. We know the result of Absorb 3. I have to draw your attention that IDA, in terms of target lesion failure, is still on the same line. It's basically a safety issue in IDA, which is shown here with a quite large difference between scaffold thrombose and stent thrombose, and they keep analyzing these results. So we know what happens with Absorb 3 IDA. There is this famous cartoon of David Capodanno. He's a master in making cartoon, and you see there is the expectation, I would say, that we've absorbed 1 and 2 of A and B. We were along the expectation. We start to diverge a little bit at absorb 3 1 year, and certainly we've absorbed 2. And then came AIDA. Chuck, any comment on that evolution?
Of the data, I think what we started to see is, as we got further into the data, was we learned that the difference in what was expected versus what we ended up seeing with Absorb boiled down to, for the first generation device, which is the Absorb GT1 device approved in Europe, boiled down to implant technique to a large degree. And the reason that the FDA approved Absorb was based on 1-year data that showed that if you exclude the small vessels, which we convinced them that we could train doctors to exclude the small vessels, the outcomes were almost spot on with Xience, you know. So the 1 year, people weren't really concerned about the 1 year. We learned how to use the first generation device for 1 year outcomes, but it was the events that started to show up at 2 and 3 years, not related to small vessels, but to larger vessels, in which the imaging that done at the time of the thrombosis showed that there was a lot of malapposition of the device relative to the vessel wall, and that probably resulted in intraluminal dismantling.
But you know, there was this famous editorial of Stefan Windecker, the device, the doctor, the technique.
Right.
Where are you now?
Yeah, I think we need both. So we were in a session this morning. I was with Tullio, and we discussed this. And it's going to require 2 things for the field of BRS, I think, to come back and be successful from everything we learned from the first generation device. One is implant technique will continue to have to be important, and so all of the PSP, but we're going 1 step further and saying that you'll need to use imaging. And in our case, of course, it would be OCT, high precise imaging, so that you not only exclude the small vessels, but you make sure you have full expansion of the scaffold and also apposition of the struts. The second piece to the success in the future, which you and I have talked a lot about and we presented back to back at CRT this year, is the technology improvements. And you've done a lot of speaking about this, and you're involved in a lot of programs that are doing this in a different way. For us, it's basically the same BBS we have always made out of PLLA, but with thinner struts, significantly thinner struts, without sacrificing radial strength and recoil, and also at the same time shortening bioresorption.
Yeah, so I'm sure Takeshi has also the same impression, because with Yoshio Numa they have looked carefully to this phenomenon of late discontinuity, which is there at 2-3 years, and seem to be associated with protrusion, thrombose, and so. Is there a critical moment there that we cannot avoid? There will be always a time of dismantling, and if the dismantling is not deeply encapsulated in tissue, it may be unpleasant.
So it's probably depend on how fast the scaffold is resorbed. I guess if you have a scaffold which resorbs faster, probably this is going to be less an issue with faster resorption.
Right. So what is fast? I mean, you will use, as everybody, the concept of 4 months, 6 months.
1 year probably.
Constrictive remodeling is gone. Now you could do whatever you want.
Probably 1 year.
Probably 1 year. Takeshi?
I'm not so sure whether the shortening of the bioresorption would lead to the solution of the intraluminal dismantling.
Sure. What is the strategy of the company?
Well, what's happening with us is by thinning the struts and reducing the total mass of the next device, the resorption will go from 36 months fully resorbed to about 26 months. So that's longer than what Tullio just mentioned, but the critical thing with polylactic acid, PLLA, is the slowness of resorption is important in a good way, in an important way, because the slower the resorption, the less the body recognizes the particulation of what's happening as degradation occurs. And that's why we went with a longer resorption rate to start with, was to reduce the inflammatory response. You get too short, start to get particulates fast enough that the body starts to recognize this, and you get an inflammatory response, at least in animals. So it may depend on the material of the scaffold,
Right.
as to whether that's important or not.
So, I mean, we have seen that that's the device-oriented cardiac event, the meta-analysis. We are behind with hazard ratio of 129 and 335. This is a cartoon of Gregstone, which is not very realistic, to be honest, because I don't see the remodeling. And we should never forget that the preparation, the pre-dilatation initially doesn't play a positive role. And the question is, if the stretching of the biodegradable is what trigger the remodeling, the expensive remodeling, and the late lumen enlargement. It makes sense to me, but we have demonstrated that one time. I think it will be tough to demonstrate that in the future. Any comment about the PSP?
Well, the two biggest impacts of PSP are excluding the small vessels. That stood out statistically. And this has been a very nice pooled analysis looking at PSP technique that Gregstone CRF published. Proper sizing to exclude the small vessels was the strongest predictor of 1-year outcomes in terms of TLF and thrombosis, mostly thrombosis. And post-dilatation with an oversized balloon, at least 18 atmospheres of pressure, was a predictor of late thrombosis after 1 year. So if you combine those two together, you get the true PSP nice combination of reducing the early events and then improving the late events.
On the other hand, this is probably just part of the problem. To come back to your question, I think it's the technique, it's the device, and it's the doctor. So I think it's a combination, an interplay of these three factors.
So being a little bit European chauvinist, I put the 5C mark. Tullio, you will bet on which one? For the time being.
Well, actually, I would like to wait for randomized trial.
You must have been educated by the Jesuit.
Yeah. Yes.
Yes, you see that?
No.
Takeshi, I mean, any
I have no idea what is good, but the important issue is very long-term outcome of the BBS bioresorbable.
Right.
So current generation DS has a very good long-term result. So we have to look at the already randomized patients for BRS versus DES for at least 10 years. So we should look at those. We could see some clinically meaningful differences.
Right. And I'm not sure that Chuck is going to say anything.
I won't say because I'm obviously conflicted, but I really like what Takeshi just said, because this is about the lifetime of the patient. What's happening in PCI today, and this is all out there, is that 80% of patients that receive PCI with any stent are alive 10 years after you do the procedure on them. That medical therapy's gotten so good, patients are living a long time with their stents. So this long-term trajectory, well beyond 1 year, and probably beyond 5 years and out to 10 years, as Takeshi says, what's really important, because I want to know if I'm getting 3 or 4 overlapping metallic stents versus a BRS, if at year 8, what I'm going to look like versus in the first year or two. But you got to be competitive early, because there's no give on safety today. The safety difference was
let's have a look about what is behind the scene. I make a collage here, so you don't have the chance to see the details, because many of them are still undercover. But I think that would draw your attention on the second one from the top left. which is from American origin of the state of Delaware, which is basically a film of polylactate, which is wrapped many times. So it's no longer it's wrapping a film around the collectors, and you can impregnate the drug. At the bottom there, left, there is a magnesium of 125 micron with tapering, only 2 connectors of polylactide, polyglycolide. Pretty interesting, good penetration. In the middle, under the title Next Generation BRS, it seems that the Japanese have succeeded to make the magnesium not permeable, so they can isolate the magnesium for 3, 4, 5 months and then get the illusion of the magnesium, and they are in the 100 micron. And then there is multiple attempts with still polylactide, either by using microfibrillar structures or macrofibrillar structures. So certainly the field is still very alive, and is just 6 different cases because I had no more space on the slide. But people are still very active, without forgetting the bioresorbable valve, which are already implanted in kids, implanted in aorta, in mitral valve. I mean, we did the control of a sheep mitral valve. Imagine bioresorbable leaflet, totally different technology. It's a macromolecule. You need enzyme and not hydrolytic system. But being optimist by nature, I think that now we have touched that field. Like the bare metal stent and the drug-eluting stent, it will take another decade, but I think that's something is going to stay.
Good.
I thank all of you for this brilliant interview with all the salient question and response to these questions. Thank you very much.
Thank you very
Thank
much.
you.